Mr. President.
All right. Well, welcome everyone to our January meeting. I hope everyone has had a happy New Year so far. It is with great pleasure we welcome back one of our long-term veterans, Max Roberts. Only now he's all grown up and he's basically a professional at the time. Oh. So this is tremendously exciting, and I know we've been trying to schedule this for a while. And well, why don't I just turn that over to Max? It's so wonderful to have you back.
Yeah, thank you everyone. My name's Max, and I actually tried to meant to give this presentation in June of last year, but then I got COVID the day of, so we had to postpone.
How dare you?
Anyway, I recently graduated from UC Santa Barbara with a degree in zoology, and I'm currently working on and off. Oh, thank you.
It's been so long.
I'm currently working on and off as a seasonal wildlife biologist, taking a gap year, and hopefully I'm going to grad school in the fall of this year, but if that doesn't work out, I'll take another gap year until it works out. But anyway, my time at UCSB was really amazing. It taught me a lot of very useful things, the courses for preparing me for my career, and had a lot of cool volunteering and work opportunities to learn about what being a biologist and a researcher is like. And it also gave me a lot of the time in my recreational life to dive further into field herping and to learn about the natural history of our local herps and all that sort of stuff. And one of my favorite things that I did while at UCSB is what I'm going to talk about today, which combined my exploratory nature with actually a research experience where I got to see what it's like to be a researcher, and that's working with the mountain yellow-legged frogs in the eastern Sierra Nevada. But before I can get into that, first have to say some basics about chytrid fungus. There's the name of the chytrid fungus, the great amphibian pathogen, mostly known as BD. And it's a kind of chytrid fungus which what it does is it disrupts the semi-permeable properties of the skin of amphibians. Some people say that it basically feeds on the skin, but other professionals say that they don't really know for sure. Research doesn't really say that. So we're just going to say it disrupts like gas exchange, water absorption, and secretion for the skin. And that's a really big deal for amphibians because, as I'm sure you're all aware, amphibians rely on these semi-permeable properties of their skin in order to breathe, to keep moist, and to secrete things. And so if they can't do that, they get really sick or can die. And so this fungus, which was only actually discovered in 1998, was essentially the black plague for amphibians because it is causing mass die-offs globally of amphibian populations, particularly of frogs and toads. It's been around, as far as we know, just under a century. But as for North America's amphibians, it's more of a dire thing because it's only arrived here in the last few decades. So there hasn't really been much of a chance for amphibians to gain immunity to this fungus, and so there's been a lot of issues with that. And so as a result, it is a great conservation concern, and there's been lots of conservation and research collaborations to try and address the effects of this pathogen. Next slide, please. And one of the groups that's actually doing a lot of important work with researching the effects of this pathogen is the Briggs Lab, University of California, Santa Barbara. And they're mainly focused on understanding disease ecology. So how disease affects the dynamics of wildlife populations. So they study a few different kinds of disease. They study sudden oak death, Lyme disease of ticks. But one of the other systems that they study is the system with chytrid and amphibians, and particularly those in California. Next slide, please. And one of the main species that they actually use to study chytrid and its effects on amphibians is actually the mountain yellow-legged frogs, which we have here in California. And there's actually two species of them currently classified. There's the Rana sierrae and Rana muscosa, both of which are found in the Sierra Nevada. Southern mountain yellow-legged frogs are also found farther south in the transverse ranges, actually the range we're in currently. But as far as sampling them and going to look for them, they look very similar and are functionally similar, so I'm just going to call them mountain yellow-legged frogs. But these are incredible amphibians when you think about what frogs are normally like. For example, the adults and juveniles will spend a lot of time just sitting on rocks and logs in the open during sunlight and during sunny weather, which is a crazy thing for a frog to do, because if you think about it, if a frog were to do that for very long, a normal frog, they would dry out, then their skin wouldn't be semi-permeable, and then they would die from that. So most amphibians couldn't do what these frogs do. They also live very long. There have been some mountain yellow-legged frogs that had pit tags in them, and they were caught 15 years after a pit tag was put in them. So that shows you just how long these frogs can live for. And also the tadpoles are extreme too. Most frogs in California, tadpoles are born and they have only a few months to a few weeks to metamorphose. And that is because their water source dries up and they have to get out of there quick. But the tadpoles here, with the mountain yellow-legged frogs, they will actually spend 2 to 3 years as tadpoles before they actually metamorphose into adults. And they're also huge, they get like this big for a tadpole, which is crazy. They're like some of the biggest tadpoles. So all of this makes this a really unique species, but the true cause of all of this, next slide please, is where they're found. And that is this habitat. The subalpine and alpine lakes of the Sierra Nevada mountains. And the thing about this habitat is, of course, it is beautiful, but it's also a really harsh environment. There's very great temperature extremes. For half the year, the lake is frozen over, and the frogs have to be underneath that for that whole period. Then in the summer, it can get hot on exposed surfaces, but usually only during the day. It's still cold even in the morning and evening. There's also harsh winds. There's rainy and snowy weather. So lots of temperature and weather extremes, and because of that, the fact that this lake is frozen over for half the year, the growing and reproductive season for these frogs is also really short comparative to ones we have maybe here in the lowlands. So that explains why they have it where they live a long time and the tadpoles take a while to grow, because their metabolism slows down for a significant part of the year. And then also, since it's so cold in the mornings and evenings, it makes sense for the adults to be on the rocks and logs basking, because then they can warm themselves, which thermoregulation for an amphibian or reptile in this kind of environment is really critical. Next slide, please. So unfortunately, the mountain yellow-legged frogs, just like many other frog populations throughout the United States, did have BD introduced to their ecosystem, and initially they were hit very hard by it. Lots of die-offs throughout the range. Many people thought that they were going to go extinct because of this fungus, because it looked like it was just going to kill them all out. However, some populations persisted. These frogs are still present in the mountains, and in some places they are doing pretty well. So it's clear that not all populations react the same way to the BD fungus. And there are actually 3 different classes of populations that have been identified. So there are declining populations. These are the ones, BD gets there and they're just not prepared for it. They just get wiped out. So either you have very few adults, because most of them have been killed off, or you have no adults. And then in that case, all you have left are the tadpoles and a few juveniles that just metamorphose and are really being hit hard and are just going to die soon. Because they can have BD on them when they're tadpoles, but they actually don't suffer the mortality until they metamorphose. So basically in that circumstance, you have it where the tadpoles turn into the tiny little frogs, and then the BD starts to take effects, like their legs don't work, and then soon they die. So that's the declining population. Stable populations are the exact opposite. BD has made it to those sites, yet the frogs are completely fine. There are lots of every life stage. They're really healthy. Even when there's BD on them, they're not sick. And then there's naive or undetermined populations where the frogs seem to be doing well. There's a lot of them, many of every life stage, but we don't know if they're actually immune to BD or what their resistance level is. Maybe BD just hasn't gotten there, and it's, if it did, that would be bad. Or it's there and we have no idea what's going on. So there's many different ways that populations are reacting to the presence of BD. And this makes one wonder, why are different populations reacting differently? Does it have to do with the environment? Are some sites more isolated? Is there little pockets in a lake in some sites where they can hide away from BD and there's less likely a chance of them getting infected? Or maybe it has to do with genetics. Maybe some of them have evolved some resistance to the fungus that other populations have. Next slide, please. And so one of the leading theories is actually that it's genetics that might be the factor that's driving this, why some populations are being affected and others aren't. But that being said, there isn't actually a lot known about this. This is just a theory. So the truth is, we don't know what the role of genetics and population resilience of these frogs is. So, yes.
Do we have any examples of populations that did decline and rebounded after the fact?
Oh yeah. That some of the stable populations, they're now stable, they're thriving.
Yeah.
Those populations had declined before, but then they came back.
Okay.
So that's why there's the indication that maybe it has to do with something with genetics and resistance. So now I got to introduce you to Monty Russell, who is a PhD candidate in the Briggs Lab, and her interest is directly involved with understanding the genetics of host-pathogen interactions. So she's currently working on a project that's in its 5th year, trying to understand the interactions between BD and amphibian populations throughout California and the role of genetics in that. So she, as you can probably guess, has worked with the mountain yellow-legged frogs, but she's also worked with the foothill yellow-legged frogs, western toads, Pacific chorus frog, the bullfrogs, many different species throughout California. So she's doing a study that's looking at a state-level assessment of the genetics of both different amphibian species and BD, and how different populations are reacting to it. And with this research, she's planning to inform amphibian conservation efforts to help us combat the effects of BD. Next slide, please. So this is where I come in, because as undergraduate at UCSB, I was looking for fieldwork opportunities involving reptiles and amphibians to gain experience with that. And just so happened that Monty was also looking for undergraduate assistants to help her with the last segment of her data collection, which was going to be sampling the frogs in the Sierra Nevada, particularly the mountain yellow-legged frogs, during the summer of 2021. So I was informed about this opportunity from a mentor and instructor of mine, and then I applied and was accepted into the position. So I got to spend, in short, 2 months in the mountains chasing these endangered amphibians, and that's what I'm going to tell you about now.
Look how skinny he is in that picture.
Yeah, it was hard work, because I will get to there. So here's a look at the crew. Here's me with long sleeves, sunglasses, and wide-brimmed hat, making sure I do not get sunburnt, because the sunburn up there is real. And then here's Imani, our graduate researcher. And then this is Kana and Iona, two other undergraduate research assistants like me that were helping her collect her data. Kana is also currently a graduate student, I believe at Oregon University in Portland, and she's doing behavioral research. Next slide, please. And then we spent 2 months in the mountains doing this research, but as you can probably figure from having to carry like big backpacks in the field to reach these remote lakes, there's no way you can do that and keep everything in your backpack for 2 months. So we had to have a base camp to stay to in between our field expeditions. And we stayed at 2 of the UC-owned reserves that were in the Sierra Nevada. First, Valentine Camp, and then the Sierra Nevada Aquatic Research Laboratory, which are located right here, basically right where Mammoth Lakes is, if anyone's been to Mammoth Lakes. So like right in the smack dab in the middle of the eastern Sierra Nevada. Next slide, please. So now I want to show a little bit of what those field stations were like. So this is, this right here is part of Valentine East Sierra Reserve. That's actually Mammoth Rock over there, which, and literally right around this is where like Mammoth Lakes is. It's like right in the center of that town. Next slide, please. Here's another view of that reserve. It has this really nice meadow here and like conifer forest, and they have like trails that go around that. So in my free time, like in between field expeditions when I need a break, I would get out to like walk and do bird watching and stuff like that. This also here, might not recognize it from this angle, but that's actually Mammoth Mountain. That's the ski resort where everyone goes to in the winter. So it's like, it's right there in Mammoth Lakes. You wouldn't think that this would be like near all that development. Here's some more looks at the habitat. This is like the edge of the meadow. They also had a stream going through there, which is kind of significant because the eastern Sierra Nevada is very dry compared to the western Sierra Nevada, because you get the rain shadow effect. Like most of the rain comes from the northwest and the west, and it hits that side of the mountains, and by the time it gets to the eastern Sierra Nevada, there's hardly any rain, so it's much drier and rockier. And so anytime that you get a stream going through, that has a lot of different plant and bird species that are just condensed in those areas, because that's the only area where there's the water and that they can be. Go back one, please. And then also another thing about that reserve is that they have these really old cabins, and that's where we say it. These are like almost pioneer level old cabins, like they've been around that long. This reserve was bought like when it was turned into a UC reserve. Before that, it was like this property where these people had these cabins, and they're just very old. So they've updated the cabins to where they have all the like amenities we need to like store food and sleep well and stuff like that. So it was very neat to be in them, but they are definitely a part of history. And so this is the cabin that we were at. Next slide, please. Also, lots of birds, as I was mentioning. This is a northern flicker that was near our cabin when we were staying there. Lots of deer too. Deer were very tame there. I don't think they're used to people, since this reserve is usually closed to the public, but they didn't.
Look, it's like, what the hell?
This one in particular was interesting because I was hiking down a different trail, and they just showed up on this trail, and he was just eating. You can actually kind of see some of the like stems that were clipped off, and yeah, he just stayed there. He didn't run at first sight. So, like, I don't know if they're used to people, but maybe just they didn't really care that much. Next slide, please. This was actually one of the most interesting birds that I saw at that reserve. It's a sooty grouse, or this pheasant that is just in like the fir and pine woodlands throughout the high mountains. They also go higher up where it's not as forested, but definitely there were a lot at Valentine Camp, so I would see them quite a few times. This one in particular on the left was a female with chicks, and so I was following them on like this log that went over, and I was looking down at them trying to get some pictures. So I'm getting near the end of this log, and next slide please. And then I hear this noise from just 15 feet away, this huge burst of an animal moving, and I look over, and it's a mother black bear and 3 cubs that just shoots out from the base of a stump. And so, and so I was like, didn't panic, but like, okay, I'm gonna turn around the log and I'm gonna go back and then give them their space. And the cubs, they just went quickly right up the tree. They knew what to do. And then the mother just looked at me well, made sure I was keeping my distance, and then she climbed the tree up there, and so I was able to get some neat shots of that. So yeah, that was my coolest bear encounter and animal encounter for the whole trip. And this is the other place that we stayed at. This was the second half of my time up there. Sierra Nevada Aquatic Research Laboratory. This is like also very close to Mammoth Lakes. It's only like a 10 to 15 minute drive out of the town, but it's like completely different because this is where the Great Basin Desert starts. It's technically not actually in the Sierra Nevada, it's like right next to it, but because it's next to it, there's this creek that comes out of the Convict Lake area. Anyone knows where that is? It's also close to Mammoth Lakes. And the creek comes out, and since it's going through the Great Basin Desert, which is usually really dry, it offers water and nutrients to like willows, aspen, and all of these trees that require the stream. So you just have this narrow strip of like stream vegetation that goes through Great Basin Desert. So it's a really interesting habitat to explore. Here's a look at what the Great Basin scrub looks like beyond the stream. This is like Sierra Nevada mountains back here, and then this is like where the Great Basin Desert starts. So it's like, well, not an immediate change, but like we were like literally right there on the transition between those two different ecoregions. And I actually did look for reptiles at both of these reserves and amphibians, but I could not find hardly any. There were species there, but for some reason they were just all really hard to find, or there weren't that many of them. So fortunately, I do not have any pictures of herps, those environments. Next slide, please. But there were a lot of birds. I'm talking about birds because birds are reptiles. So it's an endangered society. So we're gonna not leave out these very important reptile members. But there were a lot of birds there. I think when I was done, I counted like close to 70 bird species at just this second reserve, and I was only being there a month. So. Yeah, lots of bird diversity out there. This is a black-headed grosbeak. We can actually see these here in our riparian forests. And then this is a Gilbert's warbler. I think they migrate through here, but they're not common during the winter or summer. And then also again, lots of deer. They're great companions, both at the reserves and then when we went to the field.
He doesn't look as shocked to see you.
No, he walked off and was just looking back when the sun was going down. So yeah.
But were you using a real camera or a cell phone?
Yeah, I have a Nikon camera.
Yeah, because that's amazing.
Yeah. Most of my pictures and stuff here is with a Nikon camera.
The reptiles were in the ground in front of you as you're looking up.
You know, the funny thing is, he was always herping, always looking on the ground. And then when there's not anything there, you gotta find something. So he starts looking up, and now he's a birder and a herper.
Now I have to balance the two. All right, next slide, please. This was actually my favorite bird that I saw while I was there. This is the green-tailed towhee. It's a large sparrow, and they're actually a really good one to know because they are specialized to the sage scrub of the Great Basin Desert. So like you see all this vegetation in the background. It has like this kind of light green, but also grayish hue to it. And also there's like little hints of red. And then that's exactly what you see on like the green-tailed towhee. It's like a gray front, then like a smooth transitions like this nice emerald green, and then they got the red cap. So it looks obvious right here, but far away, they really do blend in because all of those colors just like blend into like the sea of like sage scrub around them. These birds are also very irritating. As a photographer, I really wanted to get a picture of them because they're one of my favorite birds. And they are very flighty and they won't let you get close. This is the only time I had one that got even close enough for this shot. And every time I would get close and scare them, they would run under a bush. I would go over to the bush where they are. I was paying attention so I knew they didn't move anywhere. So I know they're in the bush. I'd wait there for a little bit and they wouldn't move. They just sit there. And then I wouldn't get to see them, and then I'd have to just walk away because they wouldn't come out. So whenever they did that, I was so irritated. I said, okay, they're giving me the towhee treatment. So if you ever see them, you're in for the towhee treatment.
But that works. That shot was perfect.
Oh yeah, that was lucky though. That was the one that wasn't skittish.
Okay.
Like all of the others, like I couldn't get anywhere near that close.
Yeah, but you got the background perfect.
Thanks. So now, with our base camp and our home platform out of the way, let's jump off into the field expeditions. Next slide, please. So, with the very nature of the location, you know, these remote alpine and subalpine lakes and the highest mountains in the United States, you can definitely imagine that we went to some very remote sites that required a lot of backbreaking hiking up and down mountains and passes, and both on and off trail. So we did just that. We did a lot of hiking, and I would say that made up definitely the biggest portion of our work during the field expeditions, was just getting there and getting back. There was some of our trips were just day trips. It only took a day and it was pretty easy. Some trips though took longer, like 4 to 6 days. There was one trip we had planned for 9 days and we managed to squeeze it into an 8-day trip by hiking 15 miles in one day. So in these rugged mountains. This was actually that day where we did 15 miles in one day. So yeah, we got a lot of hiking experience in, and I was definitely sore at the end of those long trips. But it was worth it because you get to see these amazing landscapes. Next slide, please.
You had some oxygen adjustment that you had to go through.
Oh yeah. We actually, those base camps I pointed out are kind of lower down, like in a moderate elevation zone, like probably, you know, the higher areas in the San Gabriels here would be about where Mammoth Lakes is. And so that was a good transition zone. So I didn't really have that much altitude sickness. I just needed a day or two and then I was ready to go. And I never had altitude sickness the whole rest of the time. So yeah, if you just go to a moderate height like that, it makes it work easier. So there are two kinds of research work that we did when we went to these sites with the frogs. The first of which is to conduct visual encounter surveys or population counts. Now, these were mostly focused on the mountain yellow-legged frogs, so we were counting every adult we saw, every juvenile, just getting as good of an estimate as we could of the tadpoles. Sometimes you couldn't count them all. But then we were also keeping a note of other reptiles and amphibians too. So there were a few sites where we saw Yosemite toads. We counted any of those. We saw Pacific forest frog or a Sierra tree frog. And also we saw mountain garter snakes up there too. So if we saw any of that, we counted all of it, just to do a community population survey to know what it looked like when we took data, so that, you know, when Imani is presenting her results on the comparison between genetics and how the populations are doing, we have that full picture to look at for that summer where we got data. And for these surveys, we just went around the perimeter of these water sources, where it was a lake, pond, or a stream, and we just counted everything we saw in it and around it. And that's actually an effective way to sample for the mountain yellow-legged frogs, because in general, the juveniles and the adults are not more than 3 hops away from the water's edge. And I think one of the main reasons for that is because that's their main defensive tactic, is to hop into water. So whenever they get scared, they just go in and either dive to the bottom or go in the crack, whichever is most convenient for them. You can also see here that the habitats that we were surveying actually varied a good amount. Like, I would say this was more typical of the habitat we were surveying, like with all these bouldery stuff kind of close to above tree line. But sometimes we were lower down and we had more of this like marshy stuff that you can see over here. Next slide, please. And this is what we were looking for when we were doing these surveys. So we were just looking on all of the rocky surfaces close to the water, also logs in the ground, and seeing if we saw any frogs like this just hanging out. This is a pretty typical look at what we would find. Sometimes though, they were in the grass and they just blended in, we didn't see them, and they hopped in. But if we could hear a hop, we knew that, you know, a frog had hopped in, especially if it was a loud plunk, and we knew it was probably one of these.
So you can just count it.
Yeah,
Okay.
yeah. If we weren't sure though, we could just put a note like possible adult question mark or something.
How big were they? The like the biggest one?
So that's actually a neat thing. Thanks for bringing that up. It depends on the population. Like some populations, they were pretty small, like bat length SBL, but others they were a little bit bigger. So I think it might have to do with like just, you know, the population, maybe how high up it is. Not sure. Next slide, please. Sometimes they actually were really well hidden, and we had to keep an eye out for those.
Yeah.
Like anyone see it?
Yeah.
Yeah.
Yeah, right here. Like it looks more obvious than this because of how big this image was. But
If you're standing up, you look down, it's gonna be.
yeah, imagine this a lot smaller. Like yeah, it was not as obvious. So I took a picture of that just because of how like well blended in it was. Next slide, please. And yeah, there was no need for the arrow. So, how did we catch the frogs?
Nets.
Yes, nets is a partial answer. Nets is a partial answer. Like as you might have seen in one of the earlier images, we had nets, and yes, we did use those to catch the frogs. That's the most easiest way to be able to catch them. However, it's not really that simple, because truth is, even if you just go up to it and try to get it with a net, most of the time it would get away because they're just that fast. And as you saw earlier, where they have the lakes with like all the boulders on the shore, if they go in, they just go into the crack. Your only option is to wait for them to come back out. And so there actually were a bunch of times where we failed in trying to catch them, and we actually had to just sit there patiently, waiting for several minutes for the frogs to come out of the crack. And other times, you know, they didn't get away, but we had to be inventive. Like maybe we double team it, 2 people with 2 nets, or we had someone go on land and force someone to jump into the net we had like right below it. So we had to get really inventive with how we were catching them, and a lot of times it was really tricky. So as you can imagine, when we actually did capture them, we were very happy and excited about it. And this one in particular was my favorite capture, because this one was like right on the edge of a little rock, right near the water. If it jumped in immediately, it would have just gone in a crack. So I had an idea. What if I go around the other side of the rock, so where it can't see me, and I just sneak up, and then I just put my hands around to grab it. So I did that. I got really close. The moment of truth came, and I just put my arms around, and I was able to get it right on him, and I just grabbed it. And that was a big one too. That was a female for sure. So yeah, I was very happy about that moment. There were other times where there was one time where we were just waiting for one to come out that had gone in the water, and we had been waiting there for a few minutes. It wasn't coming, and then I just looked down and I noticed it came out, and it's like near the edge of the rocks in the water. And then my Kana, one of the assistants, was with me on like net. And so she hands me the net, and then the frog starts to swim off like into the open. So like, and then we netted them up, and then apparently it was so intriguing that like the Imani and the other assistant were like watching from a distance. And then I lifted up and like, you got it, get on land. So we got on land, and then we grabbed it, and then we took the samples. But yeah, capturing the frogs was definitely a very fun and rewarding challenge of doing the field work.
What were you catching them for? I mean, like, would you,
So that I'll get to on the next slide. So this is the start of what we were doing when we caught them. But before we took the actual samples that were really there to collect, we collected just some basic metadata information. Like weight, the sex, then SVL length, which is what is being taken here, or snout vent length. And also we took image of the frogs just to have a record of that. And then also while we are doing all of these measurements and taking the data on them, we also want to make sure that the frogs are not stressed and that they're being as healthy and we were being responsible while they're in our care. So whenever we notice their skin began to dry out, we would spray water on them to make sure that their skin isn't drying out and that they're hydrated. So we just do that a lot. As I mentioned earlier, they do sit on the rocks in the sun and they start to dry out, but at least they have the option to jump in the water if they need. So when they're in our care, we just make sure they're hydrated. And then as for the actual samples we were collecting, there were two kinds of samples that we were trying to get. So the first one is a swab sample, which is just collecting any BD fungus that is actually on their skin. So we would take Q-tip like this, and we would rub the side of the belly, inner thigh, and the bottom of the foot on both sides, 5 strokes for each spot. And every time we did that, you'd also want to apply a little bit of pressure into it to make sure you were actually getting BD in the sample if it was there. And that's just to get BD strain there, and so that later on it could be cultured in a lab and we can then study it, get the DNA from it. So that was the main, one of the 2 samples that we collected from the frogs. The other sample was a toe clipping, where we would take the longest toe that they had, just take a 2 millimeter cut of that, and that would be a tissue sample where not only we could get BD DNA, we could also get the DNA of the frogs, so we know what their DNA is like. So we're understanding both the DNA of the fungus and the DNA of the frog. And we never ran into this scenario, but if we ever did find a frog that already had toe clipping taken, we just would not use that frog in our sampling, because their toes don't unfortunately grow back. So once that's done, you don't want to do it again. Next slide, please. And then here is me doing the swabbing right here, getting ready to do the swabbing. It honestly was pretty difficult to get it in there and to do it right. These things, like, they sound pretty simple, but they're actually really precise sampling techniques that you have to use. So it was a really great experience getting to learn how to do all of that stuff.
Is the toe clipping, does that hurt them, or is it like a fingernail thing?
if it's done the right way, it does not harm them. They don't even react to it. But yeah, it definitely was something where I and the other technicians, we had to get a lot of training and advice from Amani while we were in the field, because it is a very, very sensitive and delicate procedure, and it was definitely the hardest thing we had to do with the frogs. It was definitely a challenge to do it, but by the end, by about around third or fourth expedition going out there to sample the frogs, we were all good with it and we were able to do it successfully. So yeah, when done right, it doesn't do them any harm. This also here is our setup. So we have a tray here to make sure we don't lose any samples and all of our tools. We also have these test tube boxes. When we got a sample, like a toe clip or the swab, we would put it in one of those and keep it after we're done doing sampling here out of the sun, so that we don't have the BD or grow or the toe clipping degrading because of like bacteria and stuff. Let's see what else. We also, if we had multiple frogs with us when we were doing this, we also made sure to put them under a jacket or something similar. That way they did not get a direct sunlight, because we don't want them to dry out with the heat. And we also had bags to keep them in so that they had the moisture there and that they weren't going to dry out in that way. Also, you're probably seeing this pile of mess right here, and you're wondering what that is. That is the remains of all of our gloves. So we actually use gloves to handle them because we don't want to get if there is any BD on the frog, we don't want to get that on our hands and pass it around on our stuff and bring it everywhere. And also, we had to change our gloves every time we sampled a new frog, because we don't want to transfer the fungus to other frogs if they don't have it on them. So we want to minimize our impact on these populations as much as possible. So as a result, we got a lot of trash that we had to pack out when we were done. And one final thing about the sampling is that for every site, no matter how many frogs there were, we were permitted to take a maximum of 10 frogs and sample them and then release them. So that was just the Fish and Wildlife Service. What they determined was the appropriate amount for Amani and us to sample. So if we found fewer than that number, that wasn't ideal for us because we didn't get as much data as we could, but if there were more frogs, we only sampled 10 because we're only doing the amount we need to do the research. We don't want to do more of an impact on the frogs than we have to. Next slide.
All right, yeah, and there are some questions.
Your chat has some questions.
They have a question. Was there a noticeable difference in abundance of the frogs in the marsh versus the lake habitat?
We didn't go to that many marshes, so it's hard to say for sure. But I would tend to say if I was just considering stable populations where they're doing very well, yes, there was a difference, and that might have to be with basking sites. Like if there's lots of rocks on the side of the pond, that there's more places for them to bask, and it's more efficient for them to bask and stay warm. So maybe it could mean that it's more better for them in that regard. But it might not be that too. It could be other things as well. For example, the marshy habitat I had here that was pictured earlier was a site where you can also find Yosemite toads. So it might have to deal with some interspecific competition there as well.
Okay.
So I don't really know. Those are just ideas.
All right, and then we have one more question of were the frogs pit tagged?
So we actually didn't do any work with pit tagging, but we did bring along a pit tag reader, and every frog that we sampled, we used that on it to determine whether it had been pit tagged in the past. And some of them, yes, were pit tagged. I would say most of the time though they were not. So a lot of new frogs, that's what that means. There is actually another group that does the yearly surveys of these frogs just to make sure that they're doing well. They are doing pit tagging work. So when we found frogs with pit tags in them, it was usually ones that they had done the pit tag work on. And they actually one time joined us in the field and they showed us the work that they do with the pit tags.
Were there any differences in morphology between the populations of the rocks and the populations of the wetlands? Because I know from the picture you showed, they seem to be pretty well adapted, sitting on wet rocks and stuff, so I was just curious if that kind of, their patterning and camouflage changed depending on the environment.
I definitely can't say like black and white example, like definitely with rocks they look one way, and then with muddier habitats it was different. The habitat though here with this marsh, almost all of the frogs were either lighter in color or had a more golden color like this. This one's actually from a different site, but like just imagine this, but like less of the black spotting and like just more of the golden color. So the ones at this spot are actually my favorite because they were just like bright gold a lot of the time. Whereas some of the ones with rocks, they were much darker. I might point it out if I have another picture of one from those sites. But then this one here was one that was from a rocky lake, and it was still brightly colored. So it's more complicated than just the substrate. There's other factors going on. And then this slide is just to mention, after we were done sampling, we released the frogs. It's the most important part of the job, and so I'm letting them hop back in. Because we just want to do what we can to help the frogs, and then we'll let them go and live on the rest of their lives. Next slide, please. And then we also do a lot of camping out there, because a lot of these sites mean you have to take multiple days to get to them, and then usually to do all that sampling I just mentioned would take at least half a day if you had a lot of frogs. And if you didn't have a lot of frogs, it would take even longer, because then you would have to spend time waiting to capture the frogs and searching for the frogs. So a lot of times, so we needed multiple days to do the work usually, and so lots of camping, lots of good times hanging out with the field crew, and lots of getting used to eating wet hot food with a jet boil. That was our stove for the whole trip. And to be honest, we only used it for dinner and breakfast, because lunch we almost always just had snacks. So just shoving in the carbs to keep us going up and down the trails. But then at night, that was our time to like have a warm meal. This is actually a backpacker meal there. We love those because we tried some other things that were like makeshift meals and they were not as good. So backpacker meals are better. So yeah, the food situation, definitely not the most ideal. I lost weight from the expedition, but you kind of just have to do that because you're out there. And all your food has to fit into bear cans that are like only this big, so that you don't, have it where the bear is getting in after your food. Next slide, please. Oh yes, the weather. We experienced a lot of different weather up there. It was definitely wetter during the first half of the work. Then in the later half, it was sunnier, but we also had haze come in from the fires, because 2021 there was a lot of fire activity that was happening. Like there was one in Nevada, there was one in like the Lake Tahoe area. So in the later half we had some of that haze going in. Not really to where it was like a dangerous level to work, but just a little bit you could like notice. But the most interesting weather was during the first half of the trip, because that was when it was still the wet season. And I know you don't think of June and July as a wet time in California. That's usually when it starts to really get dry. And in the lowlands it definitely does. But in the Sierra Nevada, the mountains are so high up that when the moisture, even if it's just a little bit, comes in, it gets pushed up and it condenses, and even in the summer you can get rainstorms. This day when I took this photo, it started off being a sunny day with just a few clouds, and then as the day went on, it built up to being this gray weather that you saw, and just a few minutes after I took this photo, there was a huge lightning storm, and we had to get all of our bags under a tarp and get under this, hide it out as low down as we could get during a thunderstorm, and it even started to hail too. So when I took this photo, we had hail pouring on us. So that was very interesting. We got to experience some extreme weather, and definitely made me very appreciative of just the power of the elements up there in the Sierra Nevada. If you go into those mountains, you gotta respect those mountains because lots of force up there. Next slide, please. Mountain yellow-legged frogs are also not the only creatures up there in the high mountains. There's a lot of neat wildlife up there, and it's definitely a very unique ecosystem. This is a Clark's nutcracker. Even if you are not a bird enthusiast and you're not actively trying to look for birds when you're up there, you cannot help but know this bird because they are everywhere and they are very noisy. So they make these very harsh calls. They kind of sound like a mountain crow, and yeah, you just know they're around. But even though they are very noisy, and some people find them annoying, they actually are very important in the mountains because they have a very intimate relationship with this tree you see it first in, which is the whitebark pine. They feed on a lot of different things, including mountain yellow-legged frogs. We actually saw one at one of our sampling sites while we were working actually dive in and take out mountain yellow-legged frogs. So they will eat mountain yellow-legged frogs. But their main food source is actually the pine nuts of the whitebark pine. And so every summer they stash away all these pine nuts so that they can make it through the winter where there's very little food. And so they'll stash away thousands of these nuts, and they can remember them very accurately, so they will go even when there's snow on the ground and they can't see where any of these nuts are. They will be able to accurately find them and then make it through the winter that way. But for every 1,000 seeds they bury, they forget 300. So those ones that are forgotten are then the seedlings that sprout up into new whitebark pines. So they're actually a very important seed disperser of the whitebark pine. There's also lots of mammals up there. This is a golden mantled ground squirrel. They are everywhere, like high up where the Clark's nutcrackers are, and also in the lower forests that we had to hike through to get to our sites. And some of you might be thinking that this is a chipmunk because it has that stripe on the side. And it is actually not a chipmunk. And the reason you can tell is because while it does have that side stripe, the stripe actually does not go over the eye. And all chipmunks have it where the stripe continues over the eye. So now you know how to identify a squirrel from a chipmunk. But these two things are by far my favorite mammals that we got to see up there. And the mountain yellow-legged frogs are iconic, but I think it's hard to find a more iconic mountain duo than these two. So this is the American pika. It's essentially a mountain rabbit, and they are found on these rocky talus slopes where you have just a huge amount of boulders, and they will just run across these boulders like it's nothing. They're really fast and agile. And they spend all of their summer gathering forbs, flowers, and grasses, and then creating this massive hay bale in their den underneath boulders. And then with that, they can make it through the full winter, which is 6 months or longer of just the whole thing covered with snow. So they are very busy during the summer, and they are actually the focus of a lot of conservation debate right now is being a mountain species. There's been some suggestion by researchers that with the warming temperatures of global warming, that these pika will actually move up the slopes of the Sierra Nevada until they get to the tops of the mountains, and there is no way for them to move higher. And so they may eventually, if that is the trend, be extirpated from the Sierra Nevada. So conservationists are really worried that their time in the Sierra Nevada may be limited, but more research does need to be done on that. But just wary of these little guys. The mountain yellow-legged frogs are not the only species that need our help in the Sierra Nevada. And then this guy right here is a yellow-bellied marmot. They also live on the granite slopes with the pika and feed on a lot of the same sort of things, but they seem to be more generalistic. They'll go into different habitats even lower down, so they're not necessarily as sensitive as the pika. But this guy looks like a groundhog or a woodchuck. That's because this is the relative of the groundhog or woodchuck. And they are very nice companions to have in the mountains because sometimes they'd be near our camp and they're occasionally inquisitive, and they'll just come up, like peek out from like a crack and see what you're up to. I was able to get pretty close to this guy, which is why I got the nice huge photo of him. Let's see. Another thing that I should mention about these guys is that even if you don't see them, you will know that are there because they make these huge poop piles. They have all of these scat that are like this big each pellet, and if you're in an area where they're found, you'll just find piles of it, like on the ground, little things on the rocks. So they're not very clean. They leave a lot of waste. Next slide, please. As I mentioned earlier, we also saw Yosemite toads. Not as many as the mountain yellow-legged frogs because we only went to a few sites where the toads were found, but I did get to see my very first Yosemite toads, and they are definitely a very cool species. I'm sure many of you are aware, but they're one of the few toads that's actually sexually dimorphic. This is actually a female here that has all of this like black patterning on top, and then the males, you can just imagine this without the black patterning, where it's just gold and that color. And their coloration does vary, but it's just really neat that there's that striking contrast between the sexes.
So it's only the color, not the size?
Could be the size too. I gotta be honest, I didn't really notice a difference. I only saw one male, but then when I saw a few females, it didn't look like they were that different in size. I'm sure though that there's more information about that than I'm aware. Another thing that was interesting about these is that while the mountain yellow-legged frogs were abundant like throughout our time where we were doing this research, we didn't have to worry like, oh, they're going to be less abundant later in the season where they're starting to not be as active. These we only saw them during the first half of the month, and we only saw the adults on one trip. So we were coming at the very end of the season with these guys. So the adults had just bred and they were going back to the burrows, and then all the metamorphs came out like a few weeks later. But it was just interesting that like you have these two amphibians that share the same habitats, yet one chooses to use them very differently and for a different amount of time than the other. Next slide. And then where there's frogs, yes. Next slide. There were garter snakes.
Sorry, but it doesn't like frog legs.
Yeah. I was kind of surprised that I didn't see as many snakes as I thought I was going to during this trip. There were quite a few sites we went in like nice warm sunny weather and we didn't see any snakes, which I felt that was really weird because there were frogs at those sites too. But we did go to some where there were thriving populations of frogs and we did see garter snakes. This was definitely the coolest one though, because as you can see, it was actually eating one of our study subjects, mountain yellow-legged frog. This is as found. We didn't do anything to it, and it was just holding this guy here and just waiting for it to be done so they could swallow it down. And I don't have it shown in this picture, but there were actually two other little bulges of the same size in the rest of its body.
Oh.
So it had just eaten two more of them like recently. So this garter snake was definitely doing good. For the
What was the
frogs.
approximate temperature at that time?
I don't know what the temperature was, but on the ground, probably at least in the 80s. It's like the ground temperature would be warmer than the air temperature. Yeah, we saw a few others and they were all fully alert and like doing their thing. Next slide. This one I actually got really close to because he was surprisingly tame. So when I took this photo, I was like maybe 2 feet away from him, and he sensed me, so he was like flicking his tongue. But other than that, he didn't really move at all for a while. And next up is, and here you can kind of see this is me, and like I'm only a foot away from him. He's right there. That's exactly how he was found. And we were at the site for this picture where there isn't a trail that goes right by it. So if anyone did come up there, they would be going off trail, and we didn't see any evidence that lots of people were out there. So this site sees relatively few people up there in the Sierra Nevada. So I'm guessing this thing just doesn't get into contact with many people, and it doesn't know how to react. So it just was very calm with me getting very close. But that was a really neat experience that it just sat there. Next slide, please. And I'm big on the wildlife, birds, mammals, reptiles, amphibians. That's my big thing. But the cool thing about looking for cool wildlife and doing research for cool wildlife is you get to see cool places too. So yeah, the Sierra Nevada is truly one of the most scenic areas we have in the United States. I personally think it is on par with many of the scenery places that people think of when they think of the mountains in Europe and stuff like that. And I'm not the only one who enjoys the scenery. This little guy down here also is a big fan of this home. This was my favorite shot I took the whole summer because it's just perfect. It's perfect. He's like in this little corner looking at the whole landscape. If you want to get a sense of scale of how far we traveled on some of these trips, when I took this picture, I'm at the top of a pass that we just had to hike over for 2 to 3 hours, and for the rest of the day, we hiked all the way down this into that little ravine, camped down here, and then hiked way through that and through a canyon down there. So just to give you a sense of the scale of the landscapes that we were going through. So these are some of my favorite scenery shots from the trip. So this is one of the basins that we had to go through, and there are lakes like upstream from here and also downstream, but it is kind of neat how there's actually a little bit of a stream that flows between them. And so you can actually see that it's going downhill here and then makes its way down here, and then this is the rim of the whole basin. This was actually pretty interesting. Basically, we had to go through because the site was actually not in this or down the basin. We had to get out of the basin to get to the site. So we had to go down this and then up around that corner there in order to get through to where we were going. So we actually had to do some bouldering there, and that was probably one of the scariest parts of that summer was just navigating that, because we literally were bouldering. There was no trail. It was the only time we really had to do that. But we did get to see some amazing scenery.
Were there any frogs in the flowing water areas like that? Like there were in the more stagnant areas?
So there were no frogs here in this section. Most of the populations that we saw were just in lakes, like isolated lake areas. They weren't in like streams. I know you can find some populations where they're in streams and not in the lakes, but we didn't go to any of those.
Since they have like a slower life cycle, I guess, that mean like in these areas where it could feasibly dry out faster, they wouldn't be there, or is there a different reason they're not like those stream areas?
So I don't know if it's necessarily that they could dry out. Like definitely if you have the resources right there and you're a frog just near like a pond, you're going to tend to stay there. So most frogs would end up being along like a bigger thing like a pond or a lake. But the truth is that these frogs can move between water sources. So they're not restricted to one lake. They can move like several hundred yards to potentially as much as a mile to get to other places. So they're not entirely restricted by where they are. So they could, in theory, travel down this creek to go to other ponds, but whether they actually do that, more research needs to be done on that. The Mountain Lakes crew, which I actually mentioned, the group that does yearly surveys of all the frogs in the mountains here, they actually are trying to understand that a little bit better. And I think they've partnered with some independent researchers who've been trying to figure out where they actually go. But more research needs to be done on that. Here's another landscape. You can just get a sense of how much of the mountains is just the granite rock. Like there's greening patches, but there's also just areas where it's almost entirely just the gray granite rock. So really rugged mountains. What was most interesting about this landscape actually is that it almost looks like you're looking at a little model of a mountain range, like just on a table, and someone poured some sand down the side of it. Because you have like these little sand piles that formed, but that's mostly boulders and rocks that just fell off of these mountains that kind of just piled up down at the bottom. So that gives you a sense of scale at just how big that thing is. This is actually my favorite view from all the places we went to. So I really just liked this peak, how it was like the picturesque peak you can imagine, like when you see like a little icon or image of the mountains, except lacking the snow. But this peak's called East Vedette, and this is Center Peak. They're way back in there. This is actually the Pacific Crest Trail that goes through here. So where I'm standing, and then through that canyon right there. And so we had to hike through this to get to one of our sites. And to me it was just the most scenic area that we had to go through. This is actually another picture of Center Peak, which is right over there. We were camping here on our way through, and I was just exploring around in the evening as we were getting ready to just have dinner and stuff. And we had this wonderful light coming in, illuminating like all of the lodgepole pines in the peak, and then this ominous looking background, like a storm was building. So I just took that image of the moment, just shows you again the way the clouds
You
make.
should sell a calendar.
I know, it's incredible. You really should.
36 months. There's a lot of great scenery out there. So I honestly can understand why so many people, each summer go backpacking into the Sierra Nevada. And that's actually an interesting side point to talk about too, is that over the last few years, more and more people have been doing that, where they go backpacking, and it's at a point now where you actually have to apply for a permit in advance to be able to go, just because they have so many people that want to go. And and some of these trails, like when we were going over passes to get into the Pacific Crest Trail, we couldn't go more than a few minutes going up these trails without passing multiple people. That were either coming down or that were going faster than us. There's just so many people out there, and I don't have any evidence for this, so I'm not really pointing fingers. But one of the theories that I think makes the most sense as to how BD got into the Sierra Nevada in the first place is most likely backpackers who were not cleaning their boots or shoes before going into lakes and things like that. And I should also point out, when we, as researchers, went to these sites, when we got there and when we left, we used a bleach solution on our boots and all of our equipment that got wet in the water and the mud, just to make sure that we were not transmitting the BD and pathogens to other sites.
So you mentioned the gear you used. Did you have to pay out of pocket for the gear, or did they provide the gear for you since you were applied and got into this position?
So Amani, our graduate researcher, applied for funding to have all the supplies and to house us and get the food that we would need for the trip. So I and the other assistants did not pay anything out of pocket to do this. That was all paid for by our graduate student, which is really nice.
Mhm.
She believed that we shouldn't have to pay to do research, which I agree to. What was I gonna say? Yeah, so she had to apply for grants to do that.
Okay.
So it was just an all-inclusive internship for me.
Are they still doing the internship?
So not right now. This was only one summer that she was doing all the data collection. So honestly, it was a really rare experience. And the graduate instructor who told me about this opportunity, he's also listening in right now on Zoom. He mentioned to me when he was telling me about this, because I wasn't sure if I'd be able to do it, but these opportunities don't come up a lot, and that this was really something to take seriously. So I'm very glad that he encouraged me to apply to it for sure, because they do have a lot of work going on in the Brinkus lab with different opportunities that undergraduates can get in. But as far as mountain yellow-legged frogs, I don't think they've done anything since then. But it's only a matter of time until
The next collection.
till someone else is going to do research.
You said you think it came in from hikers having poor biosecurity when they were walking through, but do you know where Chytrid like originally showed up? Like where it's endemic to, and whether or not it actually co-evolved with the amphibians in that area, or if it just kind of made the jump randomly and it just spread everywhere?
So there is a lot of debate on the origin of Chytrid in the Americas. So I definitely couldn't say like for sure, and I don't think most people would say for sure. My theory on the hikers is just a logical theory that makes sense. I actually don't. There's no evidence to show that, but you know, it just is a sensible theory. As far as how it got to the Americas, the theory that I tend to think is most probable is that it was introduced here with the African clawed frogs, which I don't know how many of you are familiar, but they are an invasive species in some parts of California, and they were used to do pregnancy tests back when.
That's how they got. They're like the most widely dispersed amphibian in the world, right now at least.
I'm not sure, but I wouldn't be surprised if they're being used for tests like that. But then when they weren't needed for the tests, lots of these labs just dumped them out into lakes here in California, and then it got in the environment. It spreads from population to population. So and then if it gets on human boots or stuff, and then people go elsewhere, then that just makes it spread faster. So I feel like that's a pretty logical idea of how it spread and got to the Americas. At least of the ones I've heard, it makes the most sense. But then again, no one really knows for sure. These are just theories.
Well, when you have the data collection all available to us and anybody else, you're going to see what the populations are doing. But we still have to just kind of guess at what's causing those things. Because even when all the data is collected and you have the data available to us, that's all you've got is the data on the populations. You don't necessarily yet know much more than that, do you? Or do you?
So yeah, with this study that Amani is doing, it's really understanding just the interplay of the genetics between frogs and toads and the fungus. It doesn't really have anything to do with like where it came from or in that regard. It's just understanding if there's any patterns between what genes are present, either the host or pathogen, or what genes are not present, and then seeing if there's any patterns there. And then if there are patterns, then maybe genetics are a part of the reason why some get infected and die and some don't get infected.
So it's like in the future, other students might come up with ideas of theories, and they might do studies to try to prove those theories, perhaps, of what's causing it and what can be done. Those are like future studies, maybe?
Oh yeah. There's a lot of research potential still in this field. Like this study that Amani is doing is like a huge study. It's like several PhDs worth of study, but even that is just scratching the surface. There's a lot of work to be done. So if anyone does want to do research with Chytrid, there's plenty of opportunities for that, and also lots of people who are willing to pay for it because of just how important it is. So just as our wrap up, thought I'd kind of address that question of how the frogs are doing. Next slide, please. So it's not a black and white answer. I can't give a satisfying answer to how the frogs are doing, but they definitely are not doing well in some places. There are declining populations where the chytrid fungus is still having a negative effect. This is one site we went to. We found several frogs like this that it clearly died from chytrid. There was one site we went to that literally like a week or two after we got up to the mountains to start data collection, the Mountain Lakes group went over there to do a survey, and they found out that BD had started a population die-off in that population, and we went there a few weeks later. It was like maybe a little over a month from when we heard that news. All the adults were gone. We couldn't find a single adult that was in that population from before the downfall, and it was just tadpoles and all these little individuals that their legs weren't working. They were clearly suffering from BD.
So you guys got data from before the die-off started?
No,
Oh.
we did not. We heard about it from the Mountain Lakes group, which they do the annual
Yeah,
surveys.
surveys.
And so
You tried to get there.
we wanted to hear back from them how the population was doing to see if it was worth it for us to sample. We ended up going, but yeah, they were not doing well. The Mountain Lakes group did a translocation project where they brought ones from a healthier population over there. So there are mountain yellow-legged frogs at that particular site, just not adults of the original population. So yes, there are populations where they're not doing well, and so BD is still a real concern, and we need to do more research and work to understand it and better protect the frogs.
Are there captive breeding programs for this specific species of frogs?
Yes, there are multiple organizations that are working with it. I know that the Oakland Zoo is one of the main breeders, and the Mountain Lakes group, they work very closely with the National Park Service and Oakland Zoo to do those captive breeding programs, and then the translocations where they'll actually take the tadpoles, get them in these nice cooled temporary storage containers with water, and then they'll helicopter them in to these sites to do
We want to
translocation.
talk about that. Was it Greg Paul or
Ian
Ian,
from the
yeah.
LA Zoo. We have a huge program at the LA Zoo for our local mountains. Not
Yeah,
for the Sierra Nevadas, but for our local mountains.
Oh, okay.
yeah, they work on the species that's the Rana muscosa, the one that's here in the transverse ranges. But it's not all bad news. In fact, I would say there's more of a positive side to this than most would think on the surface, because in many locations, these frogs are doing very well. Like there were many sites that were either naive or stable where we would go to and there would be lots of frogs. This site in particular had a lot of sandy beaches to the lake lots, the sandy shores. And you would just look down while we were doing the visual encounter survey down these beaches, and there would be all these little dots just along the edge, and that's all of these frogs just lined up down the whole thing. I think that survey we counted over 800 frogs, just adults, not including juveniles or tadpoles, that was definitely the most we'd seen at one lake. So they are doing very well in others. And as I mentioned, in some places we have found BD on the frogs and in the environment, and yet the populations are doing very well. Next two slides, please. Here's another one. We found this one had a stream coming into it. It was one of the few ones where we actually saw them using a stream, and like there were frogs all over the rocks along the stream. Next slide, please. This is a different site. There were so many tadpoles. I couldn't count all the tadpoles. I had to do an estimate of how many tadpoles there were. They were just everywhere. So these frogs are it's clearly not the end of the frogs. During the initial die-off, people were like, this is the end of the frogs. They're going to be gone soon. But no, they are still around and they are doing well in many locations. And those survivors and the ones that have been found to have resistance or supposed resistance to BD are being used in, as I was mentioning, translocation projects where they're bringing in new populations where BD has had a very negative effect and they're repopulating those lakes. Yes?
So I guess what the biggest thing about the study by Imami is that if you can figure out like a genetic profile that shows a bit more resistance, I guess they could look at the frogs that are being captive bred and see which ones have that same profile and then release those rather than ones which they know will probably fail to thrive in the environments that the chytrid fungus are just kind of sitting there.
Yes, yeah, if they can identify what genes are the ones that make the frogs more resistant to BD, or on the flip side of that, what strains of the BD chytrid fungus are more lethal to the frogs, then yes, they could do something to say which individuals would be most successful for translocation, or thinking even more into the future, is there a possibility of doing a genetic engineering project where we, transfer like that gene that's really good to helping the frogs survive into frogs and making it where these populations are more resistant. This is all like way farther in. We don't even have the results from this research yet, but those are the kinds of things that maybe these results would be able to help us down the road.
When you find a deceased frog, like several slides back on the right, were you taking samples of them also to find out whether they were deceased because of the fungi, or the fungus, or just a deceased frog?
So
we can tell that.
We weren't taking samples of them. What we did do though is if we found frogs like that, we would package them in a bag and we'd take them back to the base camps where Mountain Lakes group also works. And since they do the annual stuff with frogs and they do much more than we did on our project, we would give those samples to them and they would potentially do tests on that. But that was beyond the scope of our research. In short, there's much complexity to this. The frogs are not going to be just dying out instantly like we thought. They definitely have resistance and they are doing pretty well from a conservation standpoint. But there is more research and conservation work necessary because the species are fragile at this current point. So while they do seem pretty stable right now, that could change quickly. So that's why this research is so important. It would make it where their populations are even more stable and we can better tackle the chytrid crisis, not just with these frogs, but also potentially other populations of frogs if we do find out the role of genetics in this system. And with that being said, that is the end of my presentation. I'd be happy to take any questions.
We already asked it.
I know.
Do we have more questions?
Is there anything in the chat? Anything new?
No.
No?
So, what's the best way to look for these types of internships? And is there anyone who can apply, or are there certain restrictions?
So, I was very lucky, in short, to be able to get this experience because I will say one of the nice things about the biology department at UCSB is that they are very good at posting opportunities for internships, lab opportunities, volunteer work, even classes. They post a lot of that stuff and they post it regularly. So if you really are looking for those things, they do make that available. This, however, was not one of those things that they posted because for this position, since lots of people would want to go backpacking, working with an endangered species like this, I'm guessing there would probably have been a lot of competition among undergraduates for a position like that. So before they even, before Imani and the other members of the Briggs Lab even made this thing available, like as a general flyer, just to anyone, they were asking around if anyone had, people that they could suggest that would be good for this position. So my graduate instructor for Principles of Evolution, Ken Gilliland, he knew that I was a hardworking student. I'd come to his office hours, asked him a lot of questions about evolution, talked with him with herps, since he's big into herps. And so since he knew about my experience with field herping and enthusiasm for that, he talked to them and suggested me as a potential candidate. So I very much got this, the ability to apply for this from, you know, the connection I made there. So I did have to work for it. I, you know, interviewed and all of that. But unfortunately, this was not a public thing that just any undergraduate could apply for. It was definitely only the inner circle at that point.
But giving credit where credit is due, you and a lot of the people that aren't here tonight that we've seen so many times, a great springboard for Max to get started.
Yes.
And we owe you a great debt of gratitude and
Yes.
thankfulness
Yeah,
for
I
your
kept-
participation in this.
I kept pursuing herpetological research and studies professionally and in my recreation time from experiences I had here with the Southwest Herp Society, so I owe a lot to them.
And Mike D.
Yeah.
Oh, yes.
Everything builds.
Yeah.
Everything builds on
Yeah.
it.
And Mike D's library is, a resource center is officially opening soon too, like next
Oh,
week.
great.
Yeah,
Great.
there was an email that went around.
Oh, is that the one that the LA Zoo said they were dedicating to?
Yes, yes, it's in the grand rooms.
Oh, I know that one.
Yeah,
Mm-hmm.
so that's officially opening like next
That's
week.
awesome.
Oh,
Yeah.
that's cool.
Yeah.
Well done.
All right.
Great.
Thank you very much. Yes, and the Zoom.
Meeting is hereby adjourned.
We're gonna go have cookie and coffee.
No, it was a bit long, but it was a long.
How long?
2 months
Okay.
of research and an hour 30 minutes.
2 months straight for this.
We had like a 1 week break
Oh,
in between,
no.
but
Great.
2 months by the end.
Big chunky lady.
That's fun.
Does anybody want more coffee? Because there's some left, but I can make another pot.
Well, she's being the most calm I've seen her.
Yeah, I noticed that the whole time.
I think she just wanted a place to go sleep.
Yeah,
She can't work all day. It's like-
Oh, does she test you work with you?
Oh.
That's
Oh.
ectothermist.
Great job.
Right?
Look at this guy, he's so ambitious.